Hello, my name is Kelly Hong. Today's date is January 27th, 2020. I am presenting my portfolio PowerPoint that will be focused on the digestive system and specifically the regulation of the gastric phase of digestion. First, we will go through a quick overview of the gastric phase itself and some introductory elements to the regulation of the gastric phase. The gastric phase is the second phase of digestion, occurring after the cephalic phase and before the intestinal phase of digestion. The gastric phase of digestion takes place in the stomach, which is the organ where contents of the esophagus, known as chyme, will travel to, and from the stomach, the chyme, which becomes then a food bolus, will travel to the small intestine. The three main functions of the stomach include storage, digestion, and defense. The gastric phase accounts for approximately 2/3 of gastric secretions in the digestive system. The regulation of the gastric phase of digestion is primarily dictated by three chemicals: acetylcholine, gastrin, and histamine. These three chemicals stimulate the release of gastric acid. Conversely, a chemical called somatostatin inhibits the release of gastric acid. The vagovagal reflex is the gastrointestinal tract reflex circuit where afferent and efferent fibers of the vagus nerve coordinate responses to gut stimuli via the dorsal vagal complex located in the brain. The vagovagal reflex aids in the secretion of two chemicals that subsequently aid in gastric acid secretion. The vagal afferents of the long reflex are activated during the gastric phase of digestion by the sight, smells, and taste of food, as demonstrated in the diagram taken from Silverthorne. The preganglionic parasympathetic neurons in the vagus nerve send a signal that travels through the enteric plexus, which then causes postganglionic parasympathetic, muscarinic, and intrinsic enteric neurons to release acetylcholine, which then goes on to stimulate the target cells, the parietal and ECL cells. The parietal cells release gastric acid, and the ECL cells release histamine, which then stimulate parietal cells to release gastric acid further. A similar process occurs with the short reflexes, which are mediated through the myenteric nerve plexus located locally in the stomach. The distension of the stomach, secondary to the food bolus entering the stomach, causes stimulation of mechanical receptors in the gastric mucosa, and peptides and amino acids in the lumen of the stomach will additionally send a short reflexsensory signal to the myenteric nerve plexus to stimulate the target cells described with the long reflex, and the same chemicals will be secreted from the short reflex as with the long reflex. As described in the previous slide, acetylcholine is triggered by vagal and intramucosal reflex stimulation, which then causes gastric acid secretion and an increase in acidity in the stomach. However, the cholinergic control of gastric acid release is very complicated. Though the enteric nervous system has been identified to have some independent control that is outside the central nervous system, the full extent and details of such control has not yet been fully discovered. Therefore, certain anticholinergic drugs have been discovered to work counter to its intended purpose, such that these drugs may actually enhance gastrin release and thus gastric acid release. Gastrin is a hormone found in the bloodstream produced by enteroendocrine G cells in the pyloric glands and is one of the three chemicals that stimulates gastric acid secretion. Gastrin is stimulated by a variety of factors, including the presence of amino acids and peptides in the stomach, distension of the stomach, short neural reflexes, and even coffee. Thus, individuals with excess acid secretion syndrome are often advised to reduce or avoid coffee intake. Gastrin causes stimulation of ECL cells, which then secrete histamine, and thus indirectly stimulates parietal cells to release HCl and intrinsic factor. The third hormone that aids with the secretion of gastric acid is histamine. Histamine is secreted by ECL cells, their full name being enterochromaffin-like cells, and this occurs in the gastric glands. Histamine is a paracrine secretion and is activated by either acetylcholine or gastrin stimulation. Histamine will diffuse to parietal cells and thus stimulate gastric acid production. So although histamine is the third hormone that stimulates gastric acid secretion, it must be secondarily secreted by either acetylcholine or gastrin. Lastly, a hormone known as somatostatin, or hypothalamic growth hormone inhibiting hormone, inhibits gastric acid secretion and inhibits histamine and pepsinogen secretion indirectly. Somatostatin is stimulated in environments where the pH is less than 2, so very acidic conditions. Somatostatin is secreted by D cells in the stomach, and somatostatin functions as the primary negative feedback pathway for gastric phase secretions. This summary slide demonstrating a diagram taken from Silverthorne. As shown in the diagram, senses triggered by food, such as sight, smell, and taste, as well as trigger of the vagus nerve, will both cause stimulation of G cells, which produces gastrin. Gastrin then goes on to stimulate ECL and parietal cells, which then produce histamine and gastric acid, respectively. Histamine then also goes on to stimulate parietal cells to create more gastric acid. Once gastric acid reaches capacity, it functions as a negative feedback loop to stimulate D cells to produce somatostatin, the hormone that inhibits G cells, ECL cells, and parietal cells, effectively shutting down any further production of gastric acid. Somatostatin also inhibits chief cells, which we did not completely go over in this PowerPoint, but their normal function is to produce gastric lipase and an inactive enzyme known as pepsinogen, which then goes on to change form to become an active enzyme called pepsin. This concludes my presentation on the regulation of the gastric phase of digestion. Please find the references listed in the PowerPoint slide. Thank you for your time.